Hello everyone, my name is Ashley Esparza. I'd like to thank you for taking the time out of your day to watch my presentation. It's titled Dictyostelium, discoideum, Rab32C, Functions in Osmoregulation, Affecting the Contractile Vacuole Network During Osmotic Stress Conditions. So within our lab, what we like to do is we like to focus on vesicular transport processes, so the movement of vesicles within the cell. And we do this by focusing on their key regulators, Rab-GTPases. Okay, so Rab-GTPases, what they are is they are small proteins and are members of the Ras superfamily. Okay, and they bind guanine nucleotides, so GTPs. All right, they are considered molecular switches, and this is because they have what's called an on and an off conformation. So if you look to the image on the right, you can see that Rab, while it's attached to the membrane, is bound to GTP. This is considered its on conformation. During this conformation, it's able to actually also interact with Rab effectors, are also key in vesicular transport as well. When it is done with its function, it is hydrolyzed by the GTPase activating protein GAP. Okay, and so it'll hydrolyze the GTP, removing a phosphate, and leaving Rab in its off conformation bound to GDP. While it is in its off conformation, Rab is able to interact with what is called Rab-GDI. It's called the Rab guanine dissociation inhibitor. And what that does is that prevents Rab from prematurely binding GTP. When it is needed again, you have what's called the guanine exchange factor, GEF, and that will come and it will replace GDP with GTP, thus returning Rab to its on conformation. This as a whole is what's called the Rab cycle. So we study Rabs within a model organism called Dictyostelium discoideum. So what is Dictyostelium? What it is a small single-celled amoebozoa, okay? And it is actually found in the wild within leaf litter on the forest floor. It eats bacteria and it's a professional phagocyte. So it is used a lot in cell biology to study different cellular processes. It's also used a lot in molecular biology because it has a fully sequenced 34 megabase pair haploid genome. Okay, so it only has one pair of chromosomes, allowing it to be manipulated much easier than a more complex organism that has a diploid genome. It actually only has 6 of those chromosomes as well. Also, several of its genes are homologous to human genes, and that includes several genes that are found within human diseases. Again, it is a model organism, so it's approved for scientific studies, and it is used a lot for again, molecular and cell biology, as well as developmental studies. And this is because Dictyostelium undergoes what's called its developmental cycle. When it is starved, it will go from single-celled to multicellular, and it will create what is called a fruiting body that contains spores. This increases the organism's fitness from an ecological standpoint, ensuring that its genome will be continuing on through future generations. In order to fully understand this organism and what makes it a little different from humans is that this organism contains a very large, vast organelle called a contractile vacuole system. This contractile vacuole system actually is used for osmoregulation. Because the organism lives on the forest floor and it's very moist conditions, it needs this extra help to maintain water balance within the cytoplasm. Okay, and this we call it the CV. This CV acts as a bladder, so it's constantly pushing out and expelling this excess fluid in the cytoplasm. And it's consisted of 2 parts, a vacuolar bladder and a spongiomal network. So if you look to the image to the right, you can see these little round structures, okay, and that's the vacuolar bladder. And then these little string-like structures is the spongiomal network. Within the vacuolar bladder, it's actually responsible for the expulsion of the fluid. It is highly abundant with calmodulin. And then within the spongiomal network, it actually is where proton pumps are found, including the VH-ATPase pump, and allows for electron movement. It's also been found, because of the abundance of calmodulin, that the contractile vacuole system is responsible for calcium storage and signaling as well. So the main focus of this study is to put some emphasis on Dictyostelium's Rab32C, which is homologous to the human Rab32. So why do we want to study Rab32C and Dictyostelium? Because the human one is actually quite important, and a lot of information has recently come out regarding this protein. Within humans, it actually displays extreme tissue specificity. So what does that mean? That means it has a different function depending upon the tissue it is found in. So its function in the brain is different than its function in the skin. Okay? It has several subcellular locations. A lot of proteins are compartmentalized, so they have one location or maybe two, but this one has several. It's been found in the endoplasmic reticulum, the lysosome, endosomes, phagosomes, lysosomal related organelles. So these are organelles that are acidic like a lysosome is, but it's not the lysosome, as well as mitochondria, which is actually very rare for Rabs because no other Rab has ever been found to localize with the mitochondria. It's also one of the only Rab proteins that has the A kinase anchoring protein or AKAP ability. So what does that mean? That means it can directly tether cyclic AMP dependent protein kinase A to the membrane. Okay. Only one other Rab has been found to do that, and that's Rab38 in humans as well. Okay. And it's also found to help regulate mitochondrial associated membrane properties. So what that means is the ER has one side that faces the mitochondria and can often become stressed due to reactive oxygen species being released from the mitochondria. And so Rab32 has been found to regulate this by modulating the calcium signaling from the ER to the mitochondria. And it does this via movement and binding of calnexin. Within humans and neural tissues, it's been found to be key in development of multiple sclerosis as well as Parkinson's disease. And it is actually known to traffic the protein LRRK2 to the mitochondria within multiple sclerosis. It's key in lysosomal related organelles. Like I mentioned before, there are organelles that are acidic like lysosomes but are not lysosomes. Okay, and it's actually involved in the maturation of these organelles. So, for example, it's involved in melanogenesis, so maturation of melanosomes that are found within melanocytes, so pigmentation cells within the skin, dense granules within platelets, and phagosome maturation within macrophages. So macrophages within the immune system are professional phagocytes, and it is essential that they have phagosomes so that way they're able to phagocytize harmful substances and infected the cells as well. And again, Rab38, I mentioned it before. Rab32 is actually a paralog to Rab38, and they're extremely related in function, structure, and sequence. So what is the focus of this? Well, the overall focus of my graduate coursework is to characterize the protein Rab32C within Dictyostelium discoideum. For this presentation specifically, I'm going to focus on the work that I've done involving the contractile vacuole network within Dictyostelium. So I want to know, does Rab32C interact with the contractile vacuole network of Dictyostelium discoideum? And if it does, how does it affect the organism's osmoregulation processes? So how does it inhibit or impair or excel osmoregulation within this protozoan? Some of the the work that I began with was I wanted to look at co-localization. So does Rab32C co-localize to the contractile vacuole network? So we did this by looking at contractile vacuole marker proteins and doing immunofluorescence. So there are two marker proteins for the contractile vacuole network. First one is the VH-ATPase C subunit. So again, as I previously mentioned, the VH-ATPase pump is found abundantly within the spongiomal network of the contractile vacuole system. So I began by looking at that, and if you look, you can see the green is Rab32C overexpressor mutant. That's what OE stands for, and it is linked with a GFP tag, which glows green, utilizing fluorescent microscopy. I then used the VH-ATPase antibodies with an FITC conjugated secondary antibody, which would give the red fluorescence emission. So when put together, you can see that there are clear areas of red and there are clear areas of green. Okay, and so this suggests that there is not any co-localization within the spongiomal network aspect of the contractile vacuole system. So next, I wanted to look at calmodulin, which is found in the vacuolar bladder part of the contractile vacuole system. So again, I did immunofluorescence with anti-calmodulin antibodies. So if you look right here, you can see that there's areas of green, and here are areas of localized red, and when merged, you do see some yellow. Okay, so that suggests that there could possibly be co-localization with calmodulin within the vacuolar bladder part of the contractile vacuole system. There it is up close. So the yellow, you can see it a little clearer within that image. Next, I wanted to actually check the osmoregulation. Does the Rab32 overexpressor mutant have differences in osmoregulation during stress conditions? So first I just looked at isotonic, so just regular media, and you can see the AX2, which is our wild type parent strain, Neo, which is our empty vector, GFP only strain, and then the Rab32C overexpressor. They all show similar characteristics. And then when looked at under the fluorescent microscope, you can see that Rab32C is found localized to this area and this area as well, and scattered a little bit throughout the cell. So then we placed it in hypotonic conditions. So we placed it in straight water. We waited to see what happens. And if you look, you can see that there is exaggerated swelling. And there's also these little vacuole formations that appeared under the fluorescent microscope. It's different than the other cells. The AX2 wild type cells and the Neo were able to maintain the osmoregulation and maintain their structure when placed in water, whereas Rab32C swelled. Next, we put it in sucrose, so 0.1 molar sucrose water to create hypertonic stress conditions. And then we observe the cells under a microscope, so you can look at bright field, and you do see this thickened plasma membrane form, and it does shrink. And then you see this kind of tightening of the GFP tagged Rab32C towards the middle of the cell. Using these same conditions, I wanted to see if maybe the co-localization with calmodulin changed. So first we looked at just isotonic conditions, much like the ones previous. And you can see there is clear areas of red, clear areas of green, and when merged, clear areas of yellow. Then I looked at hypertonic, so again, sucrose water. You see this one condensed area of red, okay? And then you see this really bright area of condensed green, and there is not any overlapping within this hypertonic environment. It's very clear, green or red. Lastly, we put it in the water to do hypoosmotic stress. You see that there are areas of dark red, and areas of green, and when merged, they do overlap and create the yellow image. So one of the last things that I wanted to, one of the last things that I wanted to look at was I wanted to see if concanamycin affected the Rab32 cell line. So concanamycin is actually a drug that is used to inhibit proton pumps. So it does target the VH-ATPase pump that is found within the spongiomal network, and it inhibits it. So it prevents the energy exchange from occurring within the contractile vacuole system. I utilized 0.1 micromolar of the concanamycin and incubated the cells for 24 hours. Then I put them under these different osmotic stress for 5 minutes, along with the styral dye FM464. And the styral dye FM464 will target the contractile vacuole network within Dictyostelium discoideum. So it'll allow for visualization of the contractile vacuole system in live cells. Then I took images. So the first one you see hypoosmotic, so again water, along with the addition or incubation of concanamycin. When looked at under fluorescence, you can see that the Rab32 is very scattered. There's not a clear area of localization, whereas previously we had seen that under hypoosmotic conditions, so in water, the protein does localize quite well. If you compare it to the contractile vacuole network using the styral dye, you can see that there really isn't clear areas of co-localization. Okay, the areas where it's very bright red are distinct from the green. There are a few small parts that could be possible co-localization as depicted by the arrow. Next, I did the same thing, but without concanamycin. Right, and you can see again areas of clear localization as compared to the one above. And then if you look at the merged image, you do see that there are several areas where there is yellow, so suggesting that possibly with the addition of concanamycin, the co-localization dissipated. It was inhibited somehow. I checked just under isotonic conditions with concanamycin to see if there was a difference there because previously we did see colocalization with calmodulin, the marker protein, under isotonic conditions. You can see that there are areas of green and areas of red, but they do not overlap. So it appears as well that under isotonic conditions, concanamycin also inhibited the co-localization of Rab32C with the contractile vacuole network. There it goes. Okay. Lastly, I wanted to bring to emphasis just how exaggerated the Rab32C cells respond to hypoosmotic stress. So I took some bright field images, which you can see on the right here. After 6 minutes, you see these large vacuole formations form, and they're perfectly round, almost crater-like. In 10 minutes, the cell is blebbing and beginning to swell. By 14 minutes, the cell had burst and died, and then at 27 minutes, the cell is pretty much disintegrated. If you look at these vacuole formations under fluorescence, you can see that they're also there, and they actually align perfectly with the bright field image, suggesting that these fluorescently green vacuole formations are the same as the crater-like formation seen under bright field. All right, so that's my conclusion for this presentation. Again, so what did we learn? What did I learn? I learned that the Rab32C overexpressor cell line, it does display exaggerated swelling, vacuole formation, and increased cell death when exposed to hypoosmotic conditions. It's very exaggerated when it's put in under these conditions. When it's put in hyperosmotic conditions, it does form a thickened plasma membrane and movement of the GFP-tagged Rab32C towards the center of the cell. It colocalizes with calmodulin in both hypotonic and isotonic conditions, but not under hypertonic conditions. And you do see an altered localization upon incubation with concanamycin. And this seems to inhibit the colocalization of Rab32C with the contractile vacuole network. Some future work that I need to do is going to be to get some statistical analysis to further confirm colocalization, as well as more imaging, especially with the concanamycins under hypertonic conditions, and more immunofluorescence imaging utilizing the anti-VH ATPase subunit C antibodies under different osmotic stress conditions. I would like to thank my main advisor, Dr. Bush, my graduate committee members for all their help as well, and then individuals that have assisted me during this research endeavor: Matea Duncan, Haley Campbell, Andrew Ramirez, Azure Yarbrough, and Pooja Singh, as well as my family and friends who are always supportive. And lastly, thank you guys for the UALR Signature Experience Grant and everything that has helped contribute to the research here at UALR. And that's it. Thank you for listening.